Don't forget to put the microphone on. All right, blood. What is blood? And we're kind of starting a unit. Blood runs through where? Veins and arteries, which is part of what system? Circulatory system, or the cardiovascular system, which includes the what? The heart and the veins and arteries that carry all that stuff around, yes? So, before we can start talking about how they are transported, we have to talk about what they are transporting, and that is blood. What is blood made of? Yeah, so this diagram shows us what blood is made of. Whole blood includes liquid, which we call plasma. 55% of whole blood is plasma, so most of your whole blood is plasma. And what's most of plasma? Water. It's the least dense of the components that make up blood. The next in line is white blood cells, as far as density goes. Less than 1% of your whole blood is white blood cells. What's another name for white blood cells? Leukocytes, and also fragments of cells that float around in the circulatory system called platelets. And the other 45% of whole blood, and that's what we think of when we think of blood most, because they're very vivid and bright in color, are your what cells? Red blood cells. Or what's another fancy name for those guys? Erythrocytes. If I put these components, like a tube of blood, into something called a centrifuge and spin it around, all the heavy stuff is going to come to the bottom, and the light stuff will stay at the top. We'll do this in lab next week. And what you will see is these very specific components that separate out due to their different densities. So, when I look at the physical characteristics of blood, if I prick my finger and I bleed, what color is it? Red. It's red. Is it sticky? Or does it free flow like water? It's a little sticky, it's a little thick. You ever hear the expression, blood is thicker than water? It is. Why? Yeah, there's more than just water in it. There's some proteins which tend to be thicker in solutions. And the cells, of course. So it's a little bit sticky. The fluid, very pale, pale, pale yellow. Why do you think it's yellow? What other body fluid's yellow? Ooh, urine. Yes? You have urine in your plasma? Actually, kinda, you do. And you filter it out as waste, and you get it out of your body. And the renal system is going to filter your blood volume several different times throughout the day. Constantly. You're constantly filtering your blood volume. Because what's another thing your blood carries besides all the good stuff we keep talking about? It carries waste products. So that yellowish color that you see in plasma is the same yellowish color you see in your urine. So it's not clear. It tends to be a little on the yellowish side. What if it's really, really yellow? That could be a problem, yes? Maybe too much waste floating around in my circulatory system. Anybody ever take care of a patient with cirrhosis of the liver or hepatitis? And they actually, because of so much waste carrying in your plasma, due to problems with the other filtering systems we have in our body, the liver, you look yellow. Because blood's flowing throughout your entire body. In your skin, in your eyes. Your eye whites of your eyes, your scleras, actually look yellowish sometimes. So, What's blood for? Carry stuff. Stuff like what? Oxygen, respiratory gases. Not only oxygen, though. Carbon dioxide, too. So it carries waste respiratory gases, too. What else did you say? Electrolytes. What the heck are those? Ions. Remember way back in the chemistry chapter? Things with a what? With a charge, a positive or negative charge. And some very important ions are carried through the blood as well. And then, of course, we have the blood cells that are going to help do this and transport things. Don't forget waste products. They're being transported as well. It tends to be a little bit on the alkaline side as far as pH, but pH is kept very consistent in blood. What's pH? Go back to your chemistry chapter. Dust off some cells. Which one? What's the definition of pH? The concentration of hydrogen ions in solution. Yeah? See, you do have to remember that crap. Now, that balance has to be finely kept. That homeostasis of hydrogen ion concentrations. And we measure that concentration in pH. So what's the pH of blood? 7.35 to 7.45. That's pretty tight, isn't it? What else are we going to find in blood that's going to help keep that tight pH range going? Ever hear of a buffer? Okay, so you have a lot of buffers floating around in your circulatory system too that help maintain proper pH, and we're going to talk about those in this class as well. 8% of your body weight, and the average volume of blood, depending on how big you are, runs between 5 and 6 liters. How many gallons? 1.5 gallons. Doesn't seem like a lot, does it? It's a lot. So it's going to help perform different functions. Delivery system is basically what it does for a living. Transports oxygen from the lungs to the tissues and waste gases, carbon dioxide from the tissues to the lungs. When we talk about the respiratory system, we're going to talk about blood. Metabolic wastes from all the reactions of metabolism that's going on inside the system are also transported. Chapter 16. What else am I transporting? Hormones that will act on its target, that will change function of cells. It's also going to help maintain body temperature. Blood itself is actually a little bit higher than our core body temperature is. What's our core body temperature? 98.6 degrees Fahrenheit, 37 degrees Celsius. And blood tends to run about 100.4 degrees Fahrenheit or 38 degrees Celsius. Okay, so a little bit hotter. Well, think about the heat then has to be distributed through the body. It's got to go through muscles. You lose heat, don't you? So you have to maintain that core. So it's just a little bit hotter. It's going to help maintain normal pH by transporting all these wonderful buffers around. And transporting fluids is another very important job of blood. So all those ions we were talking about and water, extremely important. It also acts to protect you. It helps keep pressure in the system. When I talk about system, I'm talking about the cardiovascular system, so I can transport nutrients and wastes from one place to another. What if I get a hole in the circulatory system? You're just going to leak all over the place and die. No, because there are components inside blood that are going to help do what? Plug up the hole, form a clot. What other components floating around in the circulatory system might help prevent infection. Antibodies. And when we talk about some of the white blood cells or leukocytes, they might even have a direct effect on any foreign invaders that would come into the body. Macrophages, phagocytes. Macrophages don't live in the circulatory system. You know where they live? Tissues. But you know how they get there? Circulatory system. Okay? So we'll talk about those guys. So what's in blood plasma? All that stuff. Mostly water, 90% water. But the other stuff, don't disregard it, even though there's only 10%, all of that stuff is very important. So we're talking plasma proteins. Proteins that are very important to be distributed throughout the body. The biggie, the biggest component protein-wise, is something called albumin. And albumin is going to help maintain water pressure throughout the system. Make sure that water stays in the compartments it needs to stay in. Globulins, 36%. And you probably can't even see this, but it's on page 636, table 17.1. Tells us all the components. So alpha and beta and gamma globulins. Gamma globulins are those antibodies that are going to help us out, and we're going to talk about them in their own little chapter called the immune system. Proteins like fibrinogen, which are going to help do what? Plug up the hole. They float around in fluid form in blood plasma, and then series of steps activates them and turns them into a what? Turns them into a solid. Exactly. And we're going to talk about that in this chapter too. Non-protein nitrogenous substances, waste products. Nutrients. When you eat, has everybody eaten today? Who has not eaten today? Slap yourself. Your brain is not happy with you. So eat, 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 because blood is going to help transport the things you get from your food to the cells that need them for metabolism. Electrolytes, respiratory gases, hormones, all transported through blood plasma. Plasma is not like water. It's thicker than water because of all the other stuff that's in there. So we good? No, it's not like muddy water. It's like runny Jell-O. No, it's not even that thick. It's like, take the Jell-O out of the runny Jell-O, and what you have left is what's... No. A little thicker. Whole milk. Whole milk. Yeah. Okay, so that is plasma. Now, the next thing the book talks about is the cells that are running around in blood. And these are extremely important. Some of them are going to help prevent us from disease, and some of them are very important to carry what? Oxygen. As a matter of fact, they're so important for carrying oxygen, they don't even have room for a nucleus. Who are they? Red blood cells. So this is, and get familiar with this, because next week in lab, guess what you're going to be doing? You're going to figure out something called a white blood cell differential count. So you're going to count 100 white blood cells. And which ones are the white blood cells? The bigger ones with a what? With a nucleus. Lymphocyte, neutrophils, fragments of cells called platelets, monocytes. No. They are formed elements. Okay? And platelets are going to help us with the clotting process as well. There's a couple of white blood cells missing here. We're missing the eosinophils and basophils, and we'll talk about those. But this is the formed elements. So what do you have more of? White blood cells, red blood cells, or platelets? Red blood cells. So erythrocytes, or red blood cells, are basically just packages of hemoglobin. During the maturation process of this cell, before it goes into the circulatory system, it actually spits out its nucleus. So what do we end up with? If I take a side cut, normally the cell would be like this, because it's full of what? All the good stuff that cells have, and a nucleus, which is the largest of the what? Organelles. If I spit it out, see what happens? Because of that, when you look at it under the microscope, it almost looks like there's a hole there. Is there a hole there, though? No, it's just a thin area, so the light passes through it. So these guys we refer to sometimes as biconcave discs. What's bi mean? Two. Concave goes like this. It's got two of those. Yes? So erythrocytes, which are larger in number in the formed elements, are basically bags of hemoglobin. What's hemoglobin? We learned this. I'll give you a hint, and you see if you can dust off your brain cells and remember this world. It's a quaternary protein. Does that ring a bell? So it's a protein, and it's, ready? 1, 2, 3, 4 tertiary proteins put together. Yes? What are the protein globs called? They're called alpha globulin and beta globulin. So there's 4 of them. And then smack dab in the middle of each of those 4 globular proteins that make up our quaternary protein, we have this molecule. And it looks like a frisbee in there. Yes? What's this? This is heme. Hemoglobin. That's easy, right? So how many hemes do I have per hemoglobin molecule? 4. Smack dab in the middle is a what? Iron molecule. Is iron important in your diet? Why? Because you got to make hemoglobin. You don't have enough hemoglobin, you can't transport enough what? Anybody have anemia? Slightly anemic? What am I doing? You're tired a lot, yes? How come? Exactly, you're not carrying enough oxygen so you can make enough. What do I need oxygen for? Everything. No, not really. To make lots of ATP. And I need a lot of ATP. That's my energy molecule to drive my reactions of metabolism, right? So if I don't have enough hemoglobin, I don't transport enough oxygen. Guess what else hemoglobin does for a living? Besides just transport oxygen. What other respiratory gas needs transportation? Carbon dioxide. So basically, on every hemoglobin molecule, and it's kind of like an amusement park ride, there's 4 seats on this ride. Yes? And at any given time, we might have 4 passengers. When blood goes to the respiratory system, hangs out around the lungs, when you breathe in, it's going to unload its passengers, which are who? Carbon dioxide, and it's going to load up Oxygen. It's then going to take that wonderful ride through the body and go to the tissues, where it's then going to unload its oxygen and pick up carbon dioxide, yes? So at any given time, again, we can have 4 respiratory gases being transported on the hemoglobin molecule. If I have oxygens full of oxygen, they call me oxyhemoglobin. When I dump my passengers off to the tissues, guess what we call me then? Deoxyhemoglobin. And when I pick up my other passengers to bring back to the respiratory system, carbon dioxide, what do we call me? Carbaminohemoglobin. Is the hemoglobin molecule changed? No, it's just who they're carrying around. Shape changes a little, but we're not going to worry too much about that. Yes? So,
if you look under a microscope, can you tell what that cell was carrying at the time?
nope. Nope. No. Unless you do specific stains. And honestly, I don't know what those are on the top of my head, off the top of my head. You can tell when you look at these guys under the microscope, the red blood cells, whether or not they're carrying enough hemoglobin. So if I look at these cells and they have big, huge white spaces in them, that's going to be an indicator that there's not enough hemoglobin in them. If I look at them and they're really, really dense and they don't have any white spaces at all, too much. So that I can tell, but I can't tell what gases they're carrying at any given time. This is the painful part of this chapter, but it's not too bad. Where do formed elements in blood come from? Bone marrow. And guess what? All the formed elements that we're going to talk about, and we've only just talked about one, erythrocytes, all of them come from the same cell. How in the heck does that happen? Yeah, it's a stem cell. It's called a hemocytoblast. And that word blast, you've heard that before. Osteoblast. That reproduced itself, made more bone matrix, and eventually turned into osteocytes, right? Blast usually refers to some sort of immature cell. Hemo means what? Blood. So hemocyte, blood cell, immature, blast. Yes? We find this in red bone marrow that tends to hang out in the epiphysis of your bones. Remember that from last semester? And in flat bones, in that bone sandwich thing we talked about when we talked about certain flat bones. Hip bones, coxal bones, sternum. When I need more red blood cells, that cell is going to get a signal. And it's going to tell that cell it's time for you to mature into a red blood cell. So we call the Maturation Series, and are you going to have to know this? Yes. Of a red blood cell, erythro what? Erythropoiesis. Hematopoiesis is what? Is all of them, okay? But if we're talking just about red blood cells, erythropoiesis is what happens. So the hematopoietic stem cell or the hemocytoblast will get some sort of signal. Now, we just went through a chapter where we talked about signals causing cells to change. You should know the answer to this one already because you have already studied chapter 16. And you hopefully learned the name of the hormone that caused that cell to turn into a red blood cell. Anybody? What's a fancy name for a red blood cell? Erythrocytes. Guess what the name of the hormone is? Erythropoietin. Yes? And who makes erythropoietin? Good. The kidneys. So why would I need more red blood cells? Oxygen. Somebody said it. When oxygen levels start to go down. And who's gonna pick that information up? Yeah, central nervous system. There's a lot of receptors in there for a whole bunch of different things in our body, including oxygen levels. So when oxygen levels go down, that means I have to make more hemoglobin. I need to have somebody to carry that hemoglobin, red blood cells. So we'll start the maturation process of the hemocytoblast into the mature erythrocyte. Please notice, pro-erythroblast is the beginning. We go to early and late erythroblast. Then we go into normoblast. And this is where I start looking like a red blood cell. Because at this point, I'm going to start to accumulate a lot more what as I mature? Hemoglobin. What happens to that nucleus, at the normoblast stage of my maturation process? I spit it out. Am I still in the bone marrow? Yes. I'm then going to spit out the cell. The first thing we see in the circulatory system is its last phase, just before it's very mature, called the reticulocyte. And then it's going to take a little bit for that, a few hours, to become a very mature erythrocyte. It just gets reabsorbed back into the system. So it gets spit out into the bone marrow, and then you're going to break it down. It's kind of like a waste product almost. But do we recycle any of that stuff? Yeah, because there's a lot of nucleic acid in there that I can reuse. So I'm going to break it apart and reuse its parts to make more DNA and RNA and all that good stuff. Okay? So do you need to know the maturation process of a red blood cell? Yes, you do. Regulation and requirements for making red blood cells, hormonal controls, erythropoietin, and here is an example of what kind of, what's it called? Somebody said it? Negative feedback system, yes? So, the stimulus, hypoxia, low what? Oxygen. What kind of stimulus is that? Humoral, thank you very much. So, we're going to try to increase the number of red blood cells we have. We're going to signal the kidneys, get out the erythropoietin. Erythropoietin, once it eventually reaches its target, which is who? Hematopoietic stem cells or hemocytoblasts in the bone marrow. It's going to stimulate them to mature into who? Erythrocytes. They're going to get spit out into the circulatory system, and now I have the ability to carry more what? When I breathe in and out. What's going to happen to oxygen levels in the blood? They're going to rise and bring back what? Homeostasis. Yes? Other hormones might affect the production of red blood cells. And they discuss under homeostatic imbalances a little bit further. Male sex hormones, testosterone. They also, a secondary effect of testosterone is what? It's going to cause you to release more what? Erythropoietin. If I release more erythropoietin, I'm going to tend to have more what? If I'm a man, I'm going to tend to have more what? Red blood cells. Why do you think that's important for a man? Yes, tends to have larger muscle mass. I have more muscles. I need to get more oxygen to those muscles for what? to make that muscle contraction reaction and make the ATP necessary for muscle contraction. Simply, see how all this crap fits together? You gotta love it, right? So what are my important dietary requirements for proper hemoglobin production? Globins. Globins. Proteins. So proteins are important in my diet. What else? Frisbees. Iron is important in my diet. So 65% of body's iron supply is actually in hemoglobin. So most of that iron is caught up in the hemoglobin molecule. So it's very important that we get iron into our diet. And they talk about some of the manipulation of those molecules in the liver as well. So what happens, about how long does a red blood cell live? About 120 days. Another very important group, before I forget, and I don't want to decrease their importance as well, is B vitamins. Your B vitamins are extremely important in hemoglobin production. So when we talk about decreased hemoglobin and erythrocyte disorders, and what do we call a decrease in hemoglobin? It's kind of a general term. What do you have? Anemia. It can be for many different reasons, including not enough B vitamins to make my hemoglobin properly. So I can have iron deficiency anemia, not enough iron to make my hemoglobin. Or I might have a B vitamin deficiency. So what do I do with old red blood cells that have been hanging around for 120 days and they're kind of getting a little shabby? I just throw them away? Well, they don't quite get eaten. In science, we call it phagocytized. But what do I do with them? Some of it is waste, but I recycle some stuff too. So the liver has a big role in dealing with the waste products from erythrocyte destruction. Old red blood cells, get rid of those. But it also is going to help recycle some of the important things that we can use again. So this diagram shows us when we have low oxygen levels, erythropoietin, all that good stuff, new red blood cells in the system. 120 days later, we want to recycle. So the liver is going to help me. Anybody else know who else is involved in erythrocyte recycling? The spleen. There is a waste product that's going to be associated with the recycling of red blood cells. The big one is called bilirubin. And guess what color it is? Yellow. So bilirubin is picked up from the blood by the liver and secreted into the where? Intestines. Not that you've examined your stools lately, but it tends to be what color? Normally, that brownish color, yellowish color, because of the metabolism of bilirubin and some of the products, stercobilin and stercobilinogen, in your urine, in your stool. So it's important to have the raw materials to make the proper hemoglobin in the proper amount to fill up those red blood cells so we can transport oxygen and carbon dioxide through the system. If I don't have enough hemoglobin transport, and that could be for a couple of reasons. A, I don't have enough hemoglobin. B, I might not have enough what? Who transports hemoglobin? Red blood cells. Somebody mentioned earlier something about bleeding. When I bleed heavily, what happens to oxygen transport? It goes way down, right? So, different reasons for anemias. A, I might not have enough red blood cells. And that could be because I was hemorrhaging, got a hole in the circulatory system, losing a lot. or something might cause them to be bursting. You did this in lab. Osmosis and diffusion, isotonic, hypertonic, hypotonic. Is that ringing a bell?
Yep.
So the tonicity of solutions might cause some problems with your red blood cells. A hypertonic solution is going to cause your red blood cells to do what? Shrink or crenate. A hypotonic solution can cause those cells to burst or lyse. We might have something called aplastic anemia and certain drugs, and one of the big hurdles that drug companies have to overcome is its effects on bone marrow and cells of the bone marrow. You can get an awesome drug that's going to kill a tumor, bam, like that, but when you put it into humans, it might also wipe out their bone marrow. Would that be good? No, not good. So you have to go back to the drawing board. So aplastic anemia is described there as well. You might not have enough hemoglobin. Now, if you don't eat enough globins and if you don't eat enough irons, are you going to have the proper building blocks to make yourself some wonderful hemoglobin? No, you're not. So iron deficiency anemia, pernicious anemia are the results of low hemoglobin content. So a deficiency in something. Pernicious anemia is deficiency of those B vitamins we were just talking about. Abnormal hemoglobin. Hemoglobin is mostly what? Proteins, right? And who's going to determine the order in which amino acids are put together to form our proteins? Genetics, yes? Here's a scary thought. One mistake in an amino acid can drastically change the shape of that protein. This is sickle cell anemia. What's the matter with this cell? This one's cool because it looks like a fish, doesn't it? Little mouth.
Oh yeah, it's cool.
And it's got the fin over there. But that's not right. Yes? Can I carry lots of hemoglobin if I look like that? No. And this is an example of sickle cell hemoglobin because of one misplaced what? Amino acid. So one mistake in the 146 amino acids it takes to make hemoglobin, and your hemoglobin's screwed up. Can't do its job.
Is it just like for sickle cell anemia? Is it that one mistake? Like, is that typical?
That's sickle cell anemia. There's other mistakes that can cause other shape discrepancies, but that's the mistake for sickle cell anemia patients. So there's other ones, other mistakes in hemoglobin. Again, it makes it the wrong shape. So think of your amusement park ride. All right? When you get on that, what's that big round wheel that goes around? The Ferris wheel, yes? And the nice big wide seat, and you jump on the seat. Well, imagine the Ferris wheel not stopping, and then I'm going to, instead of having that nice big wide seat, boom! I'm going to make it a narrow, little, tiny, weeny seat. And you have to jump on when it's moving. Is that going to work very well? Some of you, coordinated ones, will be able to get on, but I know for sure I wouldn't be able to get on. Do you see the analogy? The wrong shape can't carry as well and can't carry as much. People who have sickle cell anemia, what do you think they do a lot? Sleep, very tired. They have trouble exerting themselves because they can't carry the oxygen that they need to. Other ones they describe here is thalassemia, which tends to be seen more in people with Mediterranean descent, Greek, Italian heritage. The globin chains are abnormally formed. Sickle cell anemia, which we just described. So, polycythemia, what's that? Too many red blood cells. Well, what's the problem with that? I can carry so much oxygen, I'll be superhuman. What happens to the consistency of your blood? Gets too thick. And when we talk about the circulatory system, we're going to talk about the tubes that carry blood around. And the thin, tiny, tiny little tubes, capillaries, what might happen if my blood starts getting too thick? They might all kind of crowd up and cause blockage. That could be a problem, yes? So polycythemia, and they talk a little bit about something called blood doping. What's that? Not dope. Yeah, some athletes got the bright idea that to try and enhance the number of red blood cells they have, it might give them more stamina in whatever sport they happen to be doing. But it can also cause a whole host of very serious problems. So polycythemia, too many red blood cells, can be a problem as well. So that's our red blood cells. Are we good? Bags of hemoglobin, anucleated, no nucleus. The next guys in line are the leukocytes, or white blood cells. And there's 5 different types of leukocytes. And I'm going to teach you a little mnemonic. You ready? Never let monkeys eat bananas. Yes? Never let monkeys eat bananas. You tell me. Percentage-wise. So never, highest percentage. Let, next in line. Monkeys, next in line. Eat bananas. So they're in order from highest to lowest. 2 categories of white blood cells, or leukocytes. This is confusing because one of them, they kind of sounds the same. We have something called granulocytes, and they're called granulocytes because of what they look like under the microscope. They look like cells with a very weird nucleus, and it looks like there's a whole bunch of sand in there, or granules. And then we have agranulocytes. Guess what? Those don't have, or have very little what? Granules. Very good. Leukocytes, some of them hang out in the circulatory system for a long time, but some of them have the ability to squeeze out of the circulatory system and go into the tissues. Remember when we talked about skin way back when, the end of last semester? And we talked about wound healing? And we talked about cells squeezing out from the circulatory system into the tissues? What was that process called? It began with a D. Diapedesis. Is that ringing a bell? Yeah. So they can squeeze out of the circulatory system, and many of them are just hitching a ride in the circulatory system. In particular, these guys down here, the monocytes. They're just kind of hitching a ride, getting from one place to another. We've already talked about him already, didn't we? When we talked about what? I just mentioned him the other day. Osteoclasts, yes? So monocytes, when they're stimulated by what? Because my calcium levels are low. Parathyroid hormone will squeeze out of the circulatory system and go hang out where? On the bones. They will become osteoclasts, and they will release bone matrix into the circulatory system. Yes? So they're just hitching a ride. They're also can squeeze out of the circulatory system and become macrophages, which are what kind of cells? Phagocytes. Help get rid of debris in the tissues. So some of them have what we call amoeboid action. They can kind of move like an amoeba. Everybody, has everybody seen an amoeba? If you haven't seen it, go Google that up. That's what white blood cells can do. They have something called positive chemotaxis. They can be attracted to signals. So in that wound healing example, when tissues are broken, the cells are kind of like cracking open an egg. They release a bunch of chemicals. And those chemicals can attract some of these macrophages to the area to help clean up a wound. Leukocytosis, what's that? Exactly. So where do leukocytes come from? Yeah, that immature cell, there it is up there. And this one's a killer, isn't it? Because there's how many different types of white blood cells? So is there the same stimulation to that cell to become a white blood cell? No, there's a whole bunch of different ones. And that is going to determine who I become when I mature. So do you need to know this maturation process? Yes, you do. So a hemocytoblast can either become a lymphoid stem cell and become who? A lymphocyte. Or it can become a myeloid stem cell and become?
Many things.
Yeah, any of those other four guys. So the production of these cells are determined by the chemicals that are being released into blood plasma. And depending on what I need and when I need it, that's when I'll release those chemicals into the circulatory system to cause the maturation process of the hemocytoblast. So, a quick look at what these guys look like. Neutrophils. We call them neutrophils because of the chemicals that are going to be picked up from the stain we use to see them under the microscope. Can we see them under the microscope if we didn't use stain? No, they'd be clear. And we wouldn't be able to determine who they are. We use a stain called Wright stain, W-R-I-G-H-T, stain. It's got a whole bunch of different things in it. Well, the neutral portions of the stain are going to be sucked up in the neutrophils granules. And the neutral portion of the stain is kind of a pinky purple color. I'm going to go to the single cells so you can see them a little bit better. Okay? So the granules, the sandy stuff in the background, are pinky purple, neutral, neutrophil. This is the nucleus. It's going to be dark purple, just like all the other nuclei we see under the microscope. It looks funny, though, doesn't it? It's kind of a weird shape, isn't it? It's not nice and round like all the other nucleuses we've seen in all our other cells so far, right? It's got a whole bunch of weird shapes. Guess what another name for a neutrophil is? You ready for this one? It's called a polymorphonuclear leukocyte. Give me a break. But it's not a bad word. Let's break down the word. What's poly mean? Many. Morpho refers to morphology, what it looks like. Polymorphonuclear, nucleus, leukocyte, white blood cell. Yes? So it's a white blood cell that has a whole bunch of different shapes to its nucleus. And all the granulocytic cells have a polymorphonuclear appearance. So how do we tell the difference between the three granulocytes? How they absorb the dye, exactly. The eosinophils, instead of having neutral colored granules, have orange ones. See all these orange little balls? That's the eosin portion of the stain. And guess what? What color are the little granules in a basophil? It's the basophilic portion of the stain. And they almost look black. They're very, very dark blue, almost black. If I didn't stain the cells, could I tell the difference between these three granulocytes? All right, we'll end there. I'll see you on... We are, by the way, we are having a quiz on this chapter Thursday. We're going to finish the chapter, yes. But we are having a quiz on this chapter Thursday. Before we go on. Where's the sign-in sheet?